There's a famous chocolate cake called a Chasseur-Torte, invented by Franz Chasseur in 1832, and it's a thing of incredible beauty. The recipe is a closely guarded secret and is passed down through the generations of the same family, so every Chasseur-Torte is made identically. The shape, the texture, the size, the taste, perfection. When one is baked at the Chasseur Hotel in Vienna, everything must be the same. The ingredients, mixing, baking, icing, every detail replicated. Change one thing, anything, and you do not have a Chasseur-Torte. You have something completely different. In a strange way, a Chasseur-Torte is like a cell. Each time it divides, its DNA is exactly copied, so each new cell is identical. So while the chef needs to understand how to make the perfect Chasseur-Torte, biologists need to know the recipe for DNA replication and its impact on every living organism on Earth. Let's eat some cake. This is Think 4. During DNA replication, there is a leading strand and a lagging strand. But how can you identify which one is which? And are they replicated differently? It's all to do with prime numbers. Not like in maths, but the prime numbers which show the position of the carbon atoms in the deoxyribose sugar. 3' and 5' are the ones we want, as you see on the nucleotide here. Nucleotides form bonds with neighbouring nucleotides, forming the sugar phosphate backbone. But when it comes to copying the DNA, there are a few problems. The strands are antiparallel. On one strand, the nucleotides run in a 5' to 3' direction, and on the other, they run in a 3' to 5'. And you know that DNA polymerase can only stick new nucleotides onto the 3' end of an existing nucleotide. So each strand is copied slightly differently, depending on which direction they go. So which strand is leading and which is lagging? You need to look at how the primers attach onto each strand. Primers are small sections of nucleic acid that attach onto the DNA strands, giving DNA polymerase a starting point for replication. DNA polymerase makes a copy of each original strand, leaving you with 2 exact copies of the original molecule by adding nucleotides onto the primers that attach to the strands. It needs free 3' ends on nucleotides to add new ones on. The leading strand is the easiest. It's a strand with a 3' end. When the primer attaches, it is going in a 5' to 3' direction, and the DNA polymerase just adds nucleotides on one at a time, continuously along the strand. The lagging strand is the original strand with the 5' end. A primer attaching to this end won't have a free 3' end for DNA polymerase to act on, as it's going in the wrong direction. So there are multiple primers on this strand, with gaps in between them, presenting lots of 3' ends for the enzyme. Once these gaps are filled, the primers are removed, and those gaps are filled until the whole strand is copied. The result? 2 exact copies of the original DNA. This process allows life to expand, organisms to grow and mature, exactly replicating their own cells again and again. The DNA of everything from humans to reindeer or rhinos, from elephants to whales, replicates through time in endless cycles. Whether you're making a Chasseur-Torte or a copy of DNA, the process must be followed in exact detail. Failure to follow the recipe and you end up with a slightly new cake or a slightly new type of cell. The process of DNA replication is complicated, but just like chocolate cake, it's essential for the continuation of life. This was Think 4. Thanks for watching.